So thus far, we've been working with weird colonial type groups, bryozoans, sponges, and then the brachiopods, our first big individual group. And we're going to talk about brachiopods as being so abundant we can actually look at populations of these things, which is then going to be our springboard to ultimately take us to looking at not just one species, but species in groups, multi-species assemblages. This is the hierarchy, and so we can talk about, look at the left column first, the individual. So you're holding an individual fossil in your hand. It's an individual organism. We say that it has an S equal to 1. So stands for species richness or number of species, 1. If you have lots of individuals of that same species, we hopefully are collecting a population. We'll talk about whether we think we have one or not. And you also then have an S of 1. But once you start talking about multi-species assemblages, which is where we're going to sort of springboard to as we start adding these different phyla to our toolkit, we start looking at communities. So communities then will have a S, or number of species, that is greater than or equal to 1, usually much greater than 1. These we're going to presume are the organisms that are living and interacting, maybe, but at least living in the same general place. And then we have a couple of bigger groupings in the paleo or ecologic hierarchy. This is the realm of biogeography, provinces, the whole biosphere, etc. And here you have lots of species, so species richness number much greater than 1. And then for the whole biosphere, well, that's counting everything. And so that's many, many, many species. Sometimes we use these terms over here on the right to describe this. So if you are actually describing the ecological characteristics of a single species that you're looking at, you would call this the paleo-ot ecology. And so what we were doing with that Waconella wacoensis, that is paleo-ot ecology. Our biology friends would just call it ot ecology. As soon as you start looking at larger groups, assemblages, associations, communities, this is called syn ecology, because some aspect of that ecology is one organism either interacting with or having something to do with the other ones. And then, as I said before, these larger categories, well, you start entering the realm of biogeography and macroevolution. Now, for all of these things, brachiopods are typically viewed as the ideal paleoecologic entities. I mentioned this in class. They have calcite mineralogy, the articulated ones at least, and it is a specific type. It's a calcite mineralogy that is very, very low in magnesium. That means it is very, very stable, especially in what we call carbonate facies in limestones. Second, their skeleton isn't like ours with 300 pieces barely articulated together. They have a simple 2-piece skeleton. And that skeleton then has a whole bunch of extensive external and internal features. We talked about some of those when we gave the description of describing the morphologies of brachiopods. So they're readily seen, and then the last bit, these things tend to be modified by their various environments in regular and predictable ways. So the rest of this slide set deals with how we can use brachiopods to talk about ancient environments and the taphonomy. And really, this is what we want to get to. We want to know if I have a sample of fossils from a community or population or anything, am I looking at what was there when these things were alive, when they were biological entities? Or has so much been removed and destroyed by taphonomic processes that I can't really say anything ecologic or biologic? Brachiopods, again, for those factors that I list right there, are ideal for giving you a measure of, okay, I have a pretty good community. I suspect that this is a good snapshot of what was there when these things were all living. Or, wow, this is a highly altered, allochthonous assemblage that has been transported, dissolved, bothered by other organisms. All I can do is say, here's what I've got. And I can't do much inference as far as biological things. So let's get started. So one of the very first things that's going to happen to a brachiopod once it dies, remember, if it doesn't have a long residence time, it's going to get buried. If that happens, then the shells are likely to still be together, still be articulated. If there is any sense of residence time, or that's extended, or then there's transport, typically what's going to happen is that the two valves will separate. They will disarticulate. And so say you are collecting somewhere, or you're looking at a slab of rocks, one thing you can do is count the number of articulated ones you see versus the number of disarticulated ones you see. And the percentage of articulated individuals that you have is, boom, a good first measure of how good your collection is, or how close it is to having been a sample of a once living population. So say I had a sample of 100 brachiopods. I picked them up, and 89% of them are still articulated, and the remaining 11% are disarticulated. That's pretty good. That's pretty good. That's a clear majority being still articulated. That's a good chance that I'm looking at a nice population. Because brachiopods have different valves, one is smaller, one is larger, they behave differently in currents. So people have done all kinds of experiments where they take different shells, the brachial valve and the pedicle valve, and remember, one is bigger, one is smaller, they have different shapes. They put them in flumes or watch them in natural conditions, and they behave differently, they move differently. So once they separate, the pedicle valves tend to go one way and the brachial valves tend to go another way, unless you capture them just after they've separated. And so what we do then is we get what's called the valve ratio. We count the number of pedicle valves versus the number of brachial valves, and in an ideal world, you have the same number. What if you find something like this? What if you say, I have 100 brachiopod specimens in my hand or in my collection. 50% of them are articulated, 50% are disarticulated. Well, okay, so we know that transport has affected these things, that they've started to separate. And then when I look at my disarticulated ones, I find that they are 40 of them are pedicle valves and 10 are brachial valves. That's a huge difference in the valve ratio. That and really you just need to count them and give the percentage of one valve is a better way to do it. Number of brachial valves, pedicle valves, if they're not equal, then you're looking at, again, more transport taking these things away. And so how unequal is a rough measure of alteration. Anytime you have any separated brachiopod valves, two sets of shells, if you have 50 of them, you should have 50 of the other one. If you don't, then something has acted to remove that other valve there. Okay. Then we measure them. We need to measure the size of everything that we can, and these sizes we roughly translate to ages. There are many different measures or things we can look at, but typically, and so by this I mean we measure every possible thing on the shell, the length, the width, the thickness, the number of the size of the ribs, the size of the deltherium. You can measure anything, but the general thing would be to measure the length, the width, the thickness. And the subtle changes in these things can have many different meanings. So there can be paleoecologic trends, there can be ecophenotypy. Remember I said that sometimes you'll have the same species in quiet environments have very different morphology than the same species in a high energy environment. If we are able to track this or collect these populations of this same species over time, like we did with Waconella wacoensis, we can say or talk about whether or not we have any evolutionary trends. And then taphonomic alteration. So we're going to look at this and see that just the measure of the size can also tell you, oh, I'm not looking at a population, I'm looking at, well, something that's an altered pile of shells. So what do we mean, first of all? Well, the mean. The mean, it's the average size measure. So typically the length, that's usually what I'll have you measure, unless you're doing research with me, in which case you'll measure lots of other things. And then a whole suite of other statistical metrics that you can use, the geometric mean, the median, the mode, etc., standard error deviation. And you're looking at a slide here of measurements of the carapace length, so the length of the skeleton of trilobites, and you see that, you know, here's your count. So here's N, so 0, 100, 200, 300. This person collected lots of shells. And then here's a bar diagram of the different sizes from 5 millimeter to 10, etc., up to some large ones, 85. Notice there's very few juveniles, very few really giant ones, and lots of them right here in the mushy middle. Distributions aren't always going to look like this, and they shouldn't. And anything that deviates from what I'm going to show you as the ideal is going to tell you that, well, okay, I've altered this community. Second set of measures is what we call the skewness. So in skew is basically, how is this different from a normal distribution? This thing here and this one up here, these are pretty much a normal distribution. There's a mean, and there's roughly an equal spread of the data on the left and right sides of the mean, of the average. It's never perfect, but the perfect one would be a nice, what we call a normal distribution. And I've showed you, you have a past test for normality. If you have something that's skewed this way, basically left skewed or right skewed. So here, this is the peak, the mean is skewed, it is moved towards the left. These things tend to have a positive value. And you've already been calculating this in these univariate summary statistics portion of past. The skewness values are given to you. In a perfect normal distribution, skewness is 0. If it is skewed to the left, it is positive. If it is skewed to the right, it is negative. So, normal data. In a normal world, there are far more juveniles, little things, than big things. This is the normal situation, especially for creatures in the ocean, where a clam might spew out enough eggs, hundreds, thousands of eggs that become little clams, but very few of them end up living to adulthood. And so when you're sampling your shells, your fossils of anything, you measure, you make a bar diagram. Past does it very nicely for us, and you look to see whether something is skewed from one side to the other. Now, there's statistical, mathematical measures of skewness, but really I just want you to eyeball these things. Is it left skewed? And this is a good sign. This would mean, all these things were born, they died, I'm sampling them. This is a good measure of a good population. If it is right skewed, way over here, then that's going to be a distribution where you have a whole bunch of adults and no juveniles. Then that means that is very biased. And sadly, this is what is expected for paleo. So what you're looking at with this diagram here is what's expected or what's found, what can be found by, say, a biologist sampling living things. He or she can go out and boom, kill everything in an area or just sample and let it go, and they can get a nice distribution like this. In paleontology, juvenile shells tend to be eaten more, they tend to be dissolved away more, they tend to be moved by transport more, and we left with a distribution that is skewed the other way. So if you have this in paleontology, if you have lots of juveniles, that's actually a good thing. There's also something called kurtosis to measure how peaked the distribution is. Again, there are some mathematical values for this. I'm not going to worry about it. But in general, these two types here, this leptokurtic versus platykurtic. Leptokurtic would be a distribution like this one here, this sort of long pointed finger. You have lots of organisms of one size and very few of the others. So even though, if you go to this other one, even though this distribution is skewed way over here, it is very pointy, and so we say that that is leptokurtic. Platykurtic is where you have small things, big things, and a whole big mushy middle here. And notice for each of these things, the mean could be exactly the same. The median could be exactly the same. You know, you could say this is, you know, average is 35.5 centimeters, millimeters, feet, whatever. But the distribution's very different. So leptokurtic is expected. Platykurtic occurs when you start getting these things altered. As I said before, the left skewed condition is normal for most things in population ecology. Lots of juveniles, very few adults. We therefore assume that ancient populations were similar. So this is ecology, and sadly, this is the normal condition for most populations in paleoecology. Think about your average vertebrate population. What if you happen to have, you know, a handful of T-Rex fossils somewhere? We tend to have lots of big adults. We have a couple of teenagers. We don't have babies, juveniles over here. So you see, paleontology is skewed this way. Juveniles get destroyed. They disappear. So you can ask yourself, do I have a good sample, meaning a good sample of a population? It should be strongly left skewed. It should also be leptokurtic. So you need to measure your fossils, eyeball the histogram, and look for deviations from this. And a sample can be anything. I deliberately put this here. This is a polished stone of some Elimia gastropods from Wyoming. And even though I don't hold the individual specimens, I can take my little calipers and I can measure that length, that length, that length, that one. There's a little one. Most of these I can't measure whole, but there's at least 6 up here where I can measure them. I can get a mean, get a distribution, and I'm probably going to say that this one would be definitely skewed in this direction here. Now these next few slides talk about these things called survivorship curves, and these are typically done more in ecology versus paleoecology, but if you suspect you have a pretty decent sample of a population, then you can run these survivorship curves. It's basically graphing the number or the percentage of survivors, what you've collected, versus the age. Now, we don't typically have the age in paleontology, but we have size, which is a rough approximation of age. Young things are small, big things are larger. But then, of course, we can count the numbers, or we can look at the percentage here. And if you notice here that they make a log scale for one of these. Now, in the old versions of PAST, they spit out these survivorship curves for you. For some reason, Øyvind Hammer, the writer, the author of PAST, has removed them from the modern version, PAST 4.04, what you have. I sent him an email, and it's coming back. So the plotting of these things is coming back He gave me a nice, a long way to make survivorship curves using what's already there, but I like the old simple thing where you just clicked one button and boom, there it was. So I'm going to present these things here, and I'm going to try to give you survivorship curves for some of the Waconella, Wacoensis data, and then ask you to sort of interpret it as well. Okay, when we look at survivorship curves, if this is sort of plotting them, we have two extremes and the middle. And these are derived from the size frequency data. So first you measure everything. Then you would generate a bar diagram like these ones that we've had or shown before. And then in a survivorship analysis, you've got the log of the number of things you've got here versus the age. And you have curves that kind of look like this one, number 1, or this one, number 2, or this one, number 3. Now, type 1 is such where you have mortality increases with age. Basically, this is what you get if you have more large or older specimens. So you'll get some curve like this, and if it is convex upward, meaning it bows out upward, it's this type 1 thing, which comes from a set of size frequency diagrams like this, where you are missing lots of the juveniles and you're heavier for these larger things. Obviously, a type 1 curve is more common for things that have been taphonomically altered. Type 2 is a relatively straight line, and this is very rare in nature. It is typically a taphonomic alteration, so notice this one here is this one is left skewed or right skewed. It's really good, but this one is kind of a distribution. You can't, it's sort of platykurtic. You can't really say what's going on. You'll get a straight line. You're almost never going to see a straight line type. Or you're going to get a type 3, which is C here, or even better, one of these ones like I've shown like this, from a distribution like these two. Lots and lots of juveniles, high juvenile mortality, and then low mortality later in life. And this is said to be convex upward. So again, I'm going to take your Waconella, Wacoensis data, where you've got the data there, you've calculated some of these things for us, the summary statistics, and I'll give you some survivorship diagrams, and we'll ask you to say whether it's type 1, type 2, type 3. Type 3 is best. That's what we want. Okay. Let's look beyond body sizes to skeletal condition. Now you're looking at two pictures of the same exact genus of brachiopod. Both of them are productids, meaning they had spines all over the body, but this one, you see the spines are still there. This one, you just barely see the little bumps, which are the bases of the spines. So when this was alive, this was shown like this. So this is just looking at the two shells and saying, this one clearly is in better shape than this one. The spines have not been broken. Here they have. So we've got several things to look at. The first is what we call epibiont coverage. When these organisms are sitting there as dead shells, so the organism has died, it is sitting on the seafloor. So there's a residence time. Don't forget that concept. Typically in the ocean environments, encrusting creatures start growing all over it. So you look at a shell and you characterize it as clean, slightly encrusted, moderately encrusted, etc., etc. And I didn't come up with these categories, and typically this is too much. I will typically ask you for epibiont coverage. Are there a lot, are there a little, or is it kind of in the mid-range? So the shell is this one here. Now if I were describing the morphology of it, I'd say, here's a big wide strophic hinge line. I see lots of very, very fine radial ornament. I see some concentric ornament. I see an interarea. There's a little bit of a deltherium here. I can identify this as a strophominid. But now look at these things growing on it. These are little bryozoans, and these little sort of cone-shaped things are little corals actually growing on it. So this shell is sitting there. Now sometimes a lot of shells will sit there and in life they get covered by epibionts. But if you have epibionts that are inside the shell, then clearly it was a dead shell that disarticulated and these things grew on it as just a dead shell. Remember in class I pointed out that brachiopods have toxic flesh, and a lot of them have little sort of places where they ooze out toxins that prevent larvae of other creatures from settling on them. So a snail can have a huge coverage of epibionts and it could still be alive and living. Brachiopods typically are only epibiont covered if they're dead. The more they have, the longer this thing has sat on the seafloor and not been buried. So it has a longer residence time. So along with all the other things we've looked at, the percentage articulated, the percentage of one valve versus another, the size frequency distribution and all of its variations, you can then look at these shells and say, ah, man, these are all covered by epibionts. They're heavily encrusted. These things had a long residence time, and therefore they've been transported. They've been through the ringer, like this. This provides direct evidence of this seafloor residence time. Again, everything I've just talked about. Sometimes people have literally taken GIS programs and mapped the epibiont locations. Basically, this is a key point up here. If you have anything on the inside of the shells, that occurred after it was dead. If it's on the outside of the shells, sometimes that indicates during a living thing, and it could vary, but we're not going to worry about that so much. Then corrosion. Corrosion is basically corrosion, dissolving, and abrasion, grinding together. And this is another measure of residence time or sedimentation rate. This is what we tried to mimic with our tumblers. So we were corroding and abrading them to see what happens to the delicate categories. Now, we didn't use brachiopods. We don't have a lot of free ones that we want to destroy like that. But you're going to look again at some of these things that we had in those tumblers all this time and talk specifically about their corrosion, their abrasion. Corrosion categories, several of these. The person that developed this has it as 1, 2, 3, 4 in Roman numerals. Basically, if you recognize something as 1, 2, or 3, either it's clean, it's uncorroded. Basically, the detail is all still there. You see all the bumps and hooks and knobs. Or it is highly corroded. The shell is smooth and ground away. Or it's sort of in the middle. Basically, most of these categories where you have more than 3, that's really hard to do. But most people can recognize the extremes. It's a good uncorroded shell, it's a highly altered corroded shell, or it's in the mushy middle. And again, I've just enlarged this one here, and look at this. So I see lots of detailed ornament, very, very fine radial ornament here, but look, there's huge portions of this shell that are smoothed off, dissolved away. Here's a little bit of epibiont coverage up here. There's some sort of destruction of the ornament over here. So this is a shell. It's not perfect, but it's not destroyed. A shell that's really gone through the ringer will have most of this gone. So this is kind of a moderate. This is in that mushy middle where it's been altered, but not in a huge, huge way. And then finally, we look at cracks, breaks, fractures. Almost all skeletal elements will break after the death of the organism. Sometimes skeletons break during the life of the organism, and then there's usually repair. If you've ever broken a bone, your bone then is always going to have a little line or a fracture place where you can see where you broke it and where it healed. And we can see this with a lot of vertebrate fossils that have bony skeletons like us. But again, if you remember our first lab, I showed you snail shells where crayfish have sort of come along and snipped and snapped on the edge of the shell, but then it grew back and corrected it. But generally, we are worried about fractures, breakages that occur after the shell has, the organism rather, and its skeleton is laying there dead. Again, the longer the residence time, the more likely you're going to break things. And we have several different types that indicate different things. So first of all, we have carinate fractures. Carinate fractures are all kinds of breaks and chips and breakage caused by just moving on the seafloor, and usually they occur along the edges of the shell. So again, look at this shell. You see a little bit is missing up here, and then compare it to this one. Here we see, well, first of all, I see corrosion. The shell is dissolved in a lot of places, but then it's chipped and broken, and it has a lot of these, what we call carinate fracture. There's a strong positive correlation with corrosion. The more corroded something is, the more carinate fractures there are. And typically, a person would just note, I have carinate fractures on my shells, but sometimes I've seen people count them, measure them, do all kinds of things. We'll see. And we'll see when we look at our results of our tumbling experiments if we have fractures as well. Then we have what are called splayed fractures. And splayed fractures happen when shells are buried, but then the weight of the sediment on top of them crushes the shell, and you get radial fractures. You get fractures, like this was been a big bulbous shell. Picture a biconvex shell, and you sit on it. You will break it and create radial fractures. These fractures radiate out in all these directions. And so these things, they might be in good shape otherwise. So this shell is in good shape otherwise. There's actually some spines sticking off of this, but it's been broken after it's buried. So even after something's buried, it can be destroyed. And this typically might indicate some high sedimentation rates. And then finally, there's a group that you're not going to have to worry about so much. Very, very rare. It's a telescoped fracture. If you think about a telescope, that old toy or whatever that kids have, you look through the tube, but you can usually sink the tube together. And that happens if things die in life position, they're dumped on by a storm or something. It's very rare. So see, I used the red robin, I didn't like my burger icon here to donate this, indicate this. It's rare, you're not going to see it. So if you do a fracture analysis, you examine all your shells of a particular species, you look for the different fracture types, you count them, and then you tally them up. Again, carinate, the bottom one here, most common. This is movement on the seafloor. Splayed, this is after it's buried, and you get these radial fractures. Telescoped, you might as well just put an X through it. I don't even have any good examples to show you. But all of these things collectively then can be used to tell me, tell you, tell your reader whether you have a sample that is a good representation, a good enough representation of an ancient original population, or it has been altered by all of these other things.